But this is Chris. This is Christian's second lecture, so please.
Okay, so thanks for joining. Today I was going to talk about a geometric view on spiking networks and distributed representations. And I invite you again to ask questions as soon as possible if there's anything that's unclear. The first half will be a bit historical, and the second half will be more about our own research. But I'll start in any case with a personal aside. That's just because I'm going to tell you a streamlined version of this research, but that's not how it started. So here's maybe more how it started initially. So last week, on Tuesday, I very briefly put up this slide. It's about a model that we built in 2005, and this model was supposed to essentially model this working memory task that I talked about, where the monkey that had to store some stimulus frequency, vibratory stimulus frequency in its short-term memory, then maintain that for 3 seconds, compare it to the second stimulus frequency, and then decide whether F1 is larger than F2. And as I showed on Tuesday, if you look at recordings in the monkey prefrontal cortex, you find these neurons that are either positively or negatively tuned to the firing rate, to the stimulus frequency that the monkey receives. So here you have time on the x-axis, firing rate on the y-axis. These different colors correspond to the different stimulus frequency F1. You see this one goes from blue to red, this one goes from red to blue. And that's because in one case, firing rate grows with stimulus frequency F1, in the other case, it drops with stimulus frequency F1. So the positive and negative correlation. And based on this data, we came up with a simple model that basically had like 2 populations of neurons. We called them plus and minus neurons, and they were coupled by mutual inhibition. And that model was able to incorporate those 3 phases of the task, loading something into short-term memory, keeping it in short-term memory, and then making a decision. Now, for keeping it in short-term memory, what the model actually creates is something that's called a continuous attractor. And what that means in practice is that, after a stimulus frequency F1 had been seen, the model was able to kind of remember it or store it in the activity of the neurons. And that's what you see here, because in the delay period, so this is a simulation from the model, in the delay period, the firing rates of the 2 populations are more or less constant. And there was one thing that I left out on Tuesday, and that is that this continuous attractor in many respects is what's called fine-tuned, meaning if you change some parameters, for instance, the precise coupling of the inhibitory connections here, then what you get is that the memory disappears. So during the delay period, the system becomes bistable, and all these different firing rates either go to, this sort of saturated firing rate is 60 hertz or a 0 hertz firing rate. So the model is very sensitive to perturbations, basically. And that has been a persistent problem with continuous attractor models. But I would say it's an even bigger problem in the field, because generally it's not something that we investigate very much when we build neural networks. We don't necessarily build neural networks and then afterwards ask, well, but is it robust if I, for instance, start knocking out neurons or changing the parameters, etc.? It's not something that's a very common thing to do. And just to give you another example, for instance, here we took, it's off the shelf, basically a recurrent neural networks, bunch of firing rate units. We train it with a particular learning algorithm. Doesn't really matter which one it is. In this case, it's forced learning from the lab of Larry Abbott. And so the network gets trained, and it gets trained to reproduce this oscillation. So this oscillation you can read out by just, applying 2 linear readouts on this recurrent network. And you see, the network reproduces a sine wave or a cosine, basically. And then we kill 5% of the neurons. And what you see, if you kill 5% of the neurons, that afterwards the oscillation stops. Okay, so that's also a system that is not robust against, killing some neurons. And truth is, we don't really know how robust the brain is, but we have some circumstantial evidence that neural circuits are pretty robust. The circumstantial evidence, for instance, is that there are things called silent strokes. So there's strokes where brain tissue gets damaged, but no one ever notices. That's why it's called a silent stroke, because there's no clear behavioral deficit that would have told you that just a bit of your brain tissue died. We also know that in diseases such as Alzheimer, at the point when you actually notice that someone has a big problem, at that point, the disease is already very much progressed, and large parts of the brain are already affected, and many cells have died. The actual onset of the disease, in terms of cells dying, etc., you don't actually notice that from the behavior of that person. Okay, so that's circumstantial evidence that the brain is robust against, for instance, the loss of neurons. So this problem interested me a lot, because precisely of this problem that I pointed out here, that in our model, it was very, very hard to make this system robust. I had to actually build a robust network model. It's in the supplementary material of this paper. And it was a lot of work, okay? It required a lot of, hacking things together. And so it was an unpleasant job of doing this. And so I was really interested in this question of robustness at that point. Then I ignored it for a while. But then in Paris, a colleague of mine, Sophie Deneve, she was working on different things, Bayesian networks, etc. But in one of the lab meetings, she showed that she had a network that had a continuous attractor, similar to the one that we had built. But she could just eliminate neurons, and it was still working. And then I said, well, that's impossible. It can't be, because we sort of know that in these kind of networks, that's not possible. Now, the network that she was using was sort of a bump attractor network. So I just suspected, it has something to do with the symmetry in your network. And if you were just to try to build the type of continuous attractor network that we had done, it's no longer going to work. But Sophie said, of course it's going to work. And she, like a couple of days back later, she came back and had a network where she showed me that, she can even build this other type of continuous attractor, and it's still robust. And I was again like, how is this even possible? People have been working on this for more than 10 years. How can it possibly be that she has a network that is robust against knocking out neurons? And so this got me really interested. And that basically then contributed or really changed part of what my lab is doing. And so now we have been working on this type of networks for, I think, by now, probably 10 years. And so that's the real reason I got interested in this type of research, okay? But now I leave that personal bit aside, and you get the streamlined version, so the hindsight of the same story, okay? And in that more streamlined version, I want to tell the story a bit different, and that is making a similar point that I made on Tuesday, which is going from single neurons to thinking about populations. So if you remember Tuesday, we talked about how people had been analyzing, one neuron at a time, mostly because that's what recording techniques allowed, and how in the last 10 years, the field has kind of switched of thinking more about the activity of neural populations and the dynamics of the state space, etc. And what I want to advocate is that maybe we need to make a similar switch in terms of the way we think about neural networks. But I'd start again first with thinking about just neural networks from the point of view of the single neuron. So the standard model, the oldest model of a neural network, and I'm going to argue a model that still very largely influences our thinking, is the following. So the idea is that each neuron is something like an analog rate unit that receives a bunch of analog inputs. These inputs are the firing rates of presynaptic neurons, X1 to X5 in this case. Then those inputs are summed on the dendritic tree that basically leads to some joint input current that is then converted in the soma of the neuron via the spiking nonlinearity into a new firing rate Y. And mathematically, you express that by saying, I'll take each input Xi, I weight it by a synaptic weight, Wi, I sum that all up, and I push it through a static nonlinearity, such as a threshold linear function, rectified linear unit, different names are sometimes used, some sigmoidal that expresses the fact that the neuron has a threshold and doesn't fire below threshold, and then there's an output firing rate y. And so why did we come up with this model, you may wonder. And I would say the evidence is, first of all, that from the very onset, from the very first recordings that people have ever done in neurons, and this is indeed the first recording, it's from Lord Adrian and Zoderman in 1926, people have observed that whenever you increase the strength of a stimulus, of a current, etc., the firing rate of a neuron will increase. And in this case, he was actually recording the toe of a cat, the sensory neuron in the toe of a cat. You have pressure in grams on the x-axis. This is the firing frequency compared to a baseline on the y-axis. And these are different experiments, okay? I kind of think it's very beautiful because, you know, they have these 4 points for like 4 different experiments, and then they draw this curve through it by hand, which of course they had to do because in 26, you know, there was no other way to do it. Couldn't send it to the computer. And so he has these several experiments, and just see, you know, as you increase the strength of the stimulus, in this case, the pressure you apply to the toe of a cat, and you record from the sensory neurons, firing rates increase, okay? More generally, and also coming from sensory systems, the idea is that neurons have a preferred stimulus. So imagine this is like an array of photoreceptors, and this is a very simplified model of a retinal ganglion cell. Okay, it's simplified because it leaves out lots of parts of the retina. So just imagine that, you know, what this retinal ganglion cell is getting is your direct input from these photoreceptors, and it follows exactly this formula, you know, linear summation of these inputs, and that is then the output firing rate of that retinal ganglion cell. Then we know mathematically that we can write this linear summation simply as the inner product between the synaptic weight vector W and the input vector X. That will be the image that falls on this spot of the screen, okay? And we know from an inner product that you can write an inner product as the cosine between those two inputs, between the input X and the synaptic weight vector W, times the length of W times the length of X. Now, if you keep X fixed in terms of length, and you just imagine that you try many, many different X, then you will see from this formula that the input here, W transpose X, is maximized when this angle is 0. So if gamma is 0, because then the cosine becomes 1. So if X aligns with W, then the input to the cell is maximized and the cell will be firing maximally. In this case, in the sort of simple, you know, on detector. And so that gives the rise to the idea that what neurons actually are, in some sense, they're feature detectors. They detect features. They start firing maximally if the input aligns with their profile of synaptic weights, okay? And that, of course, is exactly what underlies currently deep networks. That's how they're organized, right? You start with an image, then a neuron will basically take a part of the scene, maybe a square here. This will be the neuron, and if its synaptic weight vector aligns with the input, then it'll be firing maximally. And then you just iterate that procedure, you know? Then a neuron in a second layer will just do the same thing on inputs from the first layer, okay, etc. And that is, like, I would argue, the most successful computing architecture that we have at the moment. And it's really based on this idea of this standard model. And if you go through neuroscience, there are also other ideas, of course, and it's a very large field. But I would still argue that, you know, at the core of it, it's always this key idea: linear summation, static nonlinearity, okay? Now, there's a problem, though, with that standard model, and that problem predates my entry to the field. It's very old, so we know it for a long time. The problem is the following. If we believe the standard model, then if we look at what happens in a single neuron, the following things would happen. Whenever a neuron receives its preferred input, it should receive a lot of currents. Those currents should be integrated, and excitation should dominate, okay? So that's kind of shown here, like imagine the neuron receives different currents. There are excitatory currents shown in red, inhibitory currents shown in blue. And then when you add them up, you on average get a current that is excitatory. So the cell depolarizes. And as it depolarizes, the currents get integrated, the cell fires a spike, currents get integrated, cells fire a spike. And as can be shown mathematically or through simulations, the firing pattern that you should get in the cell is quite regular. Okay, so a cell that gets depolarized with a net excitatory current input, it should fire regular spikes. And that observation, I think, was first made by Softy and Koch in '94. Now, the problem is that's actually not what happens. If you look at cortical spike trains, or even spike trains in other parts of the brain, you will generally find that spikes are being fired extremely irregularly. They're kind of like similar to the random clicking of a Geiger counter. They're like Poisson processes. They seem totally random. So it's true that firing rates can go up and they can go down, but the individual spikes are not at all fired in a regular pattern. Okay. And so somehow that seems to violate the idea of the standard model that, a neuron is something like a feature detector that gets, net excitatory input when it just receives the right type of input. So what is going on? This is just another figure to show the same thing from a paper by Shedlin and Newsom, '98. That's very famous. Their question. I think it was just an interference.
Okay.
So here, they presented a motion stimulus to a neuron in area MT in the monkey, and they presented the same stimulus over and over again. So it's like 500 presentation or something, or 200 presentation of the same stimulus. And you can see that, in each of these rows here is a trial, and the spike pattern always changes a little bit. And what they did is here, they analyzed it statistically. They took a time window and counted the mean number of spikes over all of these trials, and then compared to the variance over all of these trials. And that's what's shown here. You have the mean count versus the variance. And for a Poisson process, that should go along the dashed line. And they find these dotted points. So it's not exactly a Poisson process, but it's very close. And that's an analysis you can do on many other neurons in cortex, and you'll find it's always pretty irregular. The firing is always pretty irregular. And so what's the problem with that? So one is, if you think that what the neurons are actually trying to do is they're trying to fire to signal a firing rate. So maybe they try to say, oh, this stimulus is really worth 4 spikes. Then if you had a regular rate code, you should always fire 4 spikes and there would be no problem. But in the sort of Poisson type code, where you always change the number of spikes in every trial, where in the first trial you fire 2 spikes, then 5 spikes, 2 spikes, 5 spikes, 3 spikes, etc. It always changes. So it's going to drive the downstream observer nuts because they never know what exactly you're trying to signal. And you can basically illustrate that by looking at the sort of precision that a downstream observer will have in terms of determining what stimulus was shown. So this is the spikes that you're trying to fire that are needed, and this is the spikes that are actually being fired. Then for a regular rate code, that would just be the identity. 2 spikes is what you want, 2 spikes is what you get. But if you use a Poisson spike code, then if you fire something like 8 spikes, you may get between 5 and 10 spikes or so, okay? So it's really noisy. And so that's the problem, basically. It makes the code very inefficient. So to phrase that a bit extreme, you may wonder, did evolution really converge onto such a dumb coding scheme, okay, where firing rates are conveyed via Poisson processes, which seems like not such a good idea. So many people have asked that question. It's like an old question. It's a question that actually was very much discussed at the point when I entered the field in '98. And I'm going to show you 4 ways in which people have sort of approached the question. So the first way, and that's also the way I got most excited about when I entered as a student, is it's all wrong, basically. We're really not thinking about this whole system correctly. Firing rates by itself are just an epiphenomenon of something else, okay? There was a beautiful book that came out at the time written by Bill Bialek, came out in '97, that's called Spikes, Exploring the Neural Code. And there they kind of made the point that, spikes actually are not noisy at all. We just have to look at the system the right way, and then we'll see that everything is actually super reliable, okay? So it was against this idea that there's a Poisson ratio. And here's an example of the kind of thinking, and also sort of an example of caution in terms of doing experiments. So there's a paper from Baron Koch, where again, they show, a motion stimulus that's presented here for 2 seconds. This is the onset of the stimulus, and this is a cell in area MT again. And you see every time they present the motion stimulus, the spikes fire at a different point. So there's a lot of variability. But then it turned out, what is exactly the stimulus they're using? What is a random dot motion stimulus? That means it was a bunch of dots that were moving randomly across the screen. And in each trial, well, since it's random, which dots were moving kind of changed a little bit. You always get the same sense of motion, there's always rightward motion, say, but the precise pattern of dots changed. So what they did is they said, let's use always exactly the same pattern of dots, what would be called frozen noise. And when they did that, they received a spike pattern that looked like this. So suddenly there's a lot of structure here that you didn't see before, okay? It's still somewhat, a lot of noise there, but still a lot more precision has been gained. So one idea back then was, if we just look at it the right way, we'll see that spikes are really precisely timed. And there were several ideas that came up in the 90s. So one is that only synchronous spikes matter. So that basically, we shouldn't really look at rates. The only thing that matters is that if a population of neurons fires at the same time. And the idea is that this would also give rise to the type of oscillations that you see in EEG or LFPs. So here you have a monkey that is gets a visual stimulus and is sort of looking at this orange. And when the monkey does it, you see these oscillations in the LFP, for instance. So the idea is, maybe this is what's really going on. There's an idea from the hippocampal field, especially, that what really matters are spikes in relation to some kind of ongoing oscillation. So the timing of a spike with respect to an ongoing oscillation, that is really what carries the information. So again, in this case, firing rates are just not the way things are being represented. That's called phase coding. And some of these ideas, they're still around, but none of them has led, in the last 25 years to a complete overhaul of the field, where we can say, we no longer think firing rates is what happens, it's something else. So that has not happened. I think part of the reason it hasn't happened is because these ideas have remained schematic and they have not led to some kind of satisfying network level implementation, the same way that the standard model has, okay, where you can say, here I can now build a network that works with synchronous spikes and does something interesting, or that works with phase coding and does something interesting. It just hasn't happened. Maybe it hasn't happened because not enough people have worked on these ideas. That's always one possibility. But at this point, all we can say, no, it hasn't really happened. The second route to a solution is to just ignore the problem. That has been my route, actually, for a long time, because after building a spiking network for our little, PFC to neuron model, I was very frustrated with spiking networks. And my general feeling was, I don't want to have anything to do with them. Okay, it's only when I met Sophie Deneb that I got interested again in spiking networks. Ignoring the problem, of course, is still part of the solution these days, because there's just so much we understand about firing rate networks, and they are able to reproduce certain phenomena in the brain, right? That's just the way it is. Just not all of them. So ignoring the problem is basically one possibility. And I just want to point out one way of ignoring the problem that also includes spiking network. That is just to say, well, if we have firing rate networks, we can still make them into spiking networks. So imagine you have a bunch of inputs, there's a bunch of outputs, and you want to perform some computation, X as a function of these inputs, U. But you want to use spikes for that. So there are ways of doing this. And I think the best way to do this in a principled manner is what's called the neural engineering framework, which was developed by Chris Smith and Charlie Anderson, which has actually several interesting aspects. Some of them are going to come up in the second half of this lecture. So one interesting aspect is the idea that, it's not really the neurons that matter, but it's more the idea that we can read out things over populations of neurons that matter. So they call it linear decoding, basically. And then, so that's one core principle, that you can linearly decode information. The other is that you have nonlinear encoding, the way the standard model basically has, okay? So it is in some sense the standard model, linear inputs, linear decoding, nonlinear encoding, but it gives a new twist on this idea. Then you have transformations by changing the decoding, and you can get dynamics. And I'm not going to explain how that works in detail. It's an interesting framework. That would be its own lecture. I just want to illustrate one thing that you could do with this framework, and that is to build a converter from digital to analog signals, or from analog to digital to analog. So imagine you have a bunch of digital spike trains, and we're going to think of spike trains as like binary or digital signals, where you have zeros and ones. So a linear readout is that you take those spike trains, you filter them linearly, the way Synapse does, and then you have a projection or mapping down to a lower dimensional space, and that will be the analog readouts. Now, what's nice about this way of thinking is that's exactly what we did in the data analysis on Tuesday. Okay, we take a bunch of spike trains, we filter them, and then we do a projection. So that's the way we analyze the data, okay? And it works quite well across many brain areas in terms of extracting useful information about what's represented in different parts of the brain. So that's basically a bit of a recap from Tuesday. Why linear readouts? Because they work in many different systems. You can understand E1 simple cells by thinking about linear readouts. You can understand the neurons in IT as representing a phase space. The other things you can represent, they work particularly well in motor systems. They also work, as I showed last week on Tuesday, in the prefrontal cortex and executive system. So it's a fairly general way of looking at neural data. So then you can think what a linear readout is. It's a conversion of spike trains into analog signals. And if you have such a conversion, it may seem reasonable to ask, well, how do you go back? How can you take these analog signals and then reproduce spike trains? And that is also an old question. People have thought about it in the past. And we'll think about it in the second half of the lecture again. But the classical way would be, you take those analog signals, you feed them into your neurons, okay? For instance, through a projection. So what you take is you take your analog signals, you feed them through synaptic weights into different neurons, and those different neurons then receive input currents, and each input current gets converted into a spike train. And via these spike trains, you can then basically try to decode the input signals, okay? And you can tune the parameters of such a system so that this works, so that you can take these signals and then get these reconstructed signals. All you have to tune is this weight matrix F and this decoding matrix D, basically. And what the neural engineering framework does, it tells you how to do that. It also tells you how to do computation this way, but it's just a mathematical way of basically finding the right matrices D and F to do that, okay? I'm not going to tell you how to do all the fancy stuff, but I just want to show you one demonstration of the neural engineering framework, because they use it not just to do ADA conversion, but also to perform very fancy computations. And in 2012, they had a nice paper in Science where they built a very large network model, all based on spiking neurons. It had many different areas, visual areas, executive areas such as the prefrontal cortex. It had a striatum, it had a thalamus, so it's a very large scale simulation. And they built it to make it do some tasks. And one of the tasks is shown here. So this is the visual input to the network that it gets on its retina. And you see the input would be 1, 1, 1, 1, 1, 1, then 4, 4, 4, 4, 4, 4, 5, 5, 5. And then there's a question mark as a visual input. And now what the network performs as an output, it draws with a hand. So there's a motor system. It draws 3 5s. Okay, so that's pretty impressive. And that's all done with spiking neurons. Okay, about 10 years ago. But now I just want to show you what happens if you zoom in a little bit and you look at some of those spikes. So what you're going to see is that these neurons are firing regular spikes, okay? You can see that all of these neurons are more or less firing regular spike trains. And that is because they have essentially used this idea of the standard model that you compute by having neurons being feature detectors, that detect features and they get a depolarizing input to produce an output. So that is not what we observe in biology. But it's one way of basically dealing with the problem. Okay, here's another route to the solution. That's maybe the most popular route. And that is to just assume the brain is very noisy, okay? So if you think about these signals X that I just had, and you think that, maybe they're being read out, and you generate these input currents. I just told you, if you do this, you get regular spike trains. But there's a very quick way around it, and that is you just add a lot of noise on each of these individual neurons. If you add a lot of noise, then you can now get irregular spike trains, okay? So that's a very quick way of doing this. And so you may ask, how much noise do you have to add? And the short answer is you have to add a lot of noise, okay? And that's a problem. So I'm going to save this maybe, well, let me just briefly illustrate how it works. So imagine you have basically the voltage of a neuron shown here on the x-axis. This is time, the voltage shown on the y-axis. This is time on the x-axis. And you have a net current input mu, which is shown by this black line. So if the neuron receives an input that is mu, it will not actually fire because its threshold is up here where the dashed line is. But if you now introduce a noise, illustrated here by a standard deviation sigma, then you can make the input fluctuate around that mean input mu. So even though mu does not lead the neuron to reach its threshold, by introducing the fluctuations and making them stronger and stronger, you can eventually reach the threshold and you can produce spikes. And they'll be very noisy, okay? There'll be noisy spikes. And so then the question is, you can reproduce irregular spike trains this way, but now you've got another problem, and that is you have to somehow explain where all the noise came from, okay? So you have to go back to the biophysics of single neurons, which is fairly well understood, and explain why they are so noisy. And so that's a problem because those noise sources don't exist. So I should back up. Of course, neurons are noisy. We know that that ion channels are stochastic, okay? So that, there is membrane voltage noise. We know that synapses can fail. So there is noise in the nervous system. The problem is, if you do the accounting, you see that it's totally insufficient to generate these types of Poisson spike trains. And you can also obviously see that because if you have like sensory neurons, often they're actually not random at all. They fire very regular spikes. Only if you're deep in the nervous system that everything becomes very noisy. So you could say, oh, so it's not the individual neurons. And there's a nice paper by Aldo Faisal and Daniel Wolpert, appeared in 2008, where they basically go through all of the noise sources and point this out. Okay, it's not the noise that gives you the random spike trains. It's not the noise in the neurons that gives you the random spike trains. So now the fourth solution is that if it's not the noise in the random neuron, in the individual neurons, maybe the noise comes from somewhere else. And so the idea could be, well, maybe the noise has a build up in some sense, right? Each layer is a bit more noisy and it sort of builds up. So you could think that maybe the problem is not so much that the neuron is noisy, maybe the problem is just that the neuron receives very noisy inputs. Okay, we know that if you're a cortical neuron, you receive most of your inputs from other cortical neurons. So they're all very noisy. They're all, randomly firing at Poisson spike trains. So maybe obviously because that's happening, that's why you fire randomly, okay? And this was sort of a key observation now in this influential study of Sofke and Koch in '94. And it was basically the following. If you do this, and you assume they're like, maybe 10,000 neurons here, and they're all randomly receiving inputs, and they're all independent, these random inputs, then what happens is exactly what I showed you earlier. to make that neuron fire the way in the standard model is believed, you need that the excitation is a bit stronger than the inhibition. But if excitation is stronger than inhibition, the noise actually kind of averages out. And on average, the excitation will be stronger than the inhibition. And in that case, you get a regular firing rate again, a regular spike train. So there's a self-consistency problem, because if you believe that even though all the inputs are noisy, the neuron will average out the noise and it'll produce a regular spike train. Okay, so this was a very influential study in '94, because it kind of showed that the standard model of how we think about neurons as feature detectors is inconsistent with this idea that a neuron just integrates its input. That can't be true. Now, what these authors advocated was that actually what matters are synchronous spikes here. But that got a lot of attention, and many people were unhappy with that solution. And it led to an alternative idea of how you can get very noisy spike trains, which is that you could also get this if you just make sure that on average, excitation and inhibition are balanced. So if you have like excitatory current, and on average, the inhibitory currents are exactly the same size, then when you subtract them, you kind of subtract out to zero, and only the small fluctuations will remain. And then you're in a state now where basically you actually don't have a net depolarizing current, but you just have like a net current that's zero, and then there are fluctuations, and those fluctuations is what drives the neurons to fire spikes once in a while. And there was an influential set of studies from Mike Shadlin, Bill Newsom, Van Weeswijk and Sompolinsky. There's also Nicolas Brunel and Daniel Amit that then showed how you can make this work, okay? And that led to the idea of balanced network. So it's possible if you assume that excitation and inhibition are balanced. And that was a very influential, because then people started studying balanced networks. It's still a problem though, because it did not rescue the standard model, okay? It showed you how you can generate noise. It showed how you can generate irregular and asynchronous spike trains without having to assume that all the individual neurons are noise. So it showed you how to generate noise at the network level. It did not explain why you would want to generate so much noise, okay? And so to drive that message home and put it a little bit in a cartoonish way, we can say that even now we're a field that whenever we want to do something that is functional, whenever we want to do some kind of computation, the go-to model are firing rate networks, okay? Because these are highly functional, but they produce unrealistic spiking activity, okay? And many spiking networks that you will see these days that are trying to produce function too, they all often fall into the same trap, basically, that they try to emulate firing rate networks in different ways, but they often don't generate this sort of realistic balanced spiking activity. On the other hand, if you want to stay in a biologically realistic regime, and the regime of balanced networks, then now you can get your realistic firing activity, but you don't know how to compute with these networks, okay? And one thing I should say though is that balanced networks haven't just been very influential because, I forgot to say this, I should really say this. They haven't just been influential because they produce irregular activity. Rather, what people have done is they have gone into neurons and tried to measure whether they actually receive balanced input. And lo and behold, that's exactly what people found. So they found that neurons receive, on average, input that balances excitation and inhibition. That's, I think, one of the main reasons that people are very interested in balanced networks, because they do seem to capture something about what the brain does. It just seems to not agree totally with our standard model of the feature detector. So before the break, I want to maybe take 2 steps back and ask, why does the brain produce spikes in the first place? And maybe I'll just want to ask that to the students. What do you think? Or what do you think why the brain uses spikes? Why do neural circuits use spikes? Yeah.
Is that for information transmission, to transmit signals very far? And
So what is
also, I have also a second point. I think it's like, I think that neurons that are connected, but they communicate through spikes, are kind of like independent and connected together. They can perform computations, and when they reach some decision, they send a spike. While if they were continuously connected, they would influence each other throughout the whole time.
Right. So so that's a good. Those are two good points. So the first point is maybe it's about long-range communication, and I think that's a very good point, because it's true that long-range communication, if you just use voltages, for instance, so analog signals, doesn't work very well, because axons are very small, and so in that case, noise does actually build up. Okay, so if a very long axon, you're trying to transmit just a membrane voltage over millimeters, nothing is going to arrive at the end except noise. Okay, and so we can say that spikes are a solution to that problem of transmitting signals over longer distances. The second point you made was more about the computation. So there it's more complicated, because it turns out that there are organisms that actually do not use spikes. So one classical case is C. elegans. 10 years ago, it was believed that it doesn't produce any spikes. The story has changed a little bit now. So spikes have been found, but I think it's still mostly true that this is an organism that has 302 neurons. So it's a worm. And most of these neurons do not produce any spikes. They all communicate just with membrane voltages. Okay. Another example that's maybe closer to home is the retina. So in the retina, if you so the light comes from this side and then hits the photoreceptor layer, and the photoreceptor cells, they do not produce any spikes. Okay, they communicate with a whole bunch of neurons in this intermediate layer here, horizontal cells, bipolar cells, etc. None of them produce any spikes. Okay, it's only the retinal ganglion cells at the output of the retina, whose axons then go all the way to the thalamus, that actually do produce spikes. So there's a lot of computation in the retina. It's all analog. Okay, so that's sort of circumstantial evidence for the fact that whenever neurons don't really need long distance communication, so maybe they don't actually spike. Okay. Here's a more engineering perspective on the general problem and how to compute and how computation interfaces with energy. There's a very beautiful paper by Raul Zapeška in '98, where these ideas are from, and it works as follows. So if we compare, so what he did actually, which is sort of an interesting computation, he said, okay, how many bits per second does the brain compute with? And so he makes sort of a back of the envelope calculation, looking at the number of neurons, number of synapses in the human brain, and he comes up with some crazy number. Okay, I don't know what it is, terabits per second or something like that. And then he says, well, I'm an engineer. If I now was to build a computer that basically has the same computational power as the brain, let's imagine I can do that. How much energy does this computer need? And what he comes up with is that the computer basically needs something like a nuclear power station. So it has the energy needs of a whole city in some sense. Okay, that's a computer built on current digital hardware that has the same computational power as the brain. And maybe he got it wrong by a factor of 10, but with this orders of magnitude difference, it doesn't really matter. It's just a huge difference. Whereas what does the brain use? Well, the brain uses something like 12 watts. It's like a dim light bulb. Okay. So there's a huge difference in how efficiently we compute with digital computers and how efficiently the brain computes. That, of course, has also been figured out by the hardware industry for some time. And there's a whole field called neuromorphic engineering that is sort of built on the idea of trying to build systems that are more energy efficient and still can allow us to do computations. So the idea of Raul Zapeška was the following. So the first idea is that analog computation is actually very energy efficient. Because what is analog computation? Analog computation is the idea to compute with the laws of physics, okay? So if you have a digital computer and you want to add two numbers, you have a bunch of zeros and ones, and then there's an algorithm with which you have to add those numbers, very complicated, and then you have added the two numbers. It's a very complicated floating point algorithm that allows you to add two numbers. If you have an analog computer, you just take two wires, you put them together, the laws of physics are going to add the currents that go to those two wires. So it's basically just physics. So you can easily add using the laws of physics. And that's what happens in a neuron, right? You have like currents that flow in the dendritic tree. And if it's a passive dendritic tree, everything just gets added automatically. It doesn't require complicated floating point operations. The laws of physics do the addition for you. So that's what makes analog computation very efficient. Of course, engineers have known that for a while. The problem with analog computation is always that how to contain the noise, okay? Because noise keeps building up. So it's very hard to build an analog computer. That's why all our computers are digital, because digital computation has the huge advantage that it's completely noise-free, but it has the big disadvantage that it's very energy wasteful. So the idea of Ralsar-Peshka was, well, maybe the brain is trying to have the best of the two worlds. It's trying to be energy efficient by using, maybe analog computations and the dendritic trees and the somas of neurons, etc. But it also wants to have the noise contained by, having maybe signal transmission and maybe also digital signal restoration using spikes. Okay, sort of just an engineering argument. You didn't actually say, how you're supposed to do this. You just said maybe that's the way the whole system works, and that's why it is so energy efficient. Using analog computation inside the neurons, and then just using the spikes as a way of transmitting the signal down to the next area if you want, okay? And so in the second part of my lecture, I then want to give you some ideas of how something like this could work. But I would make the suggestion to maybe have a 5-minute break now.
Christian,
Yeah.
just one question about these last comments. The reason that it's useful against noise, the digital, is that it's kind of the amplitude is noiseless, right? So you have 0 or 1, but that doesn't talk at all about the timing. So this argument. So this argument is a static argument, doesn't include the dynamical part, right?
It's true. It's true. The timing part is missing here. So in digital computation, of course, you have like a synchronized clock. That's what you mean, right?
Right.
And I'm not arguing that actually. And this is mostly just about the idea of restoring signals, basically. But it's static. It's a static argument. Okay. Also, this argument that Ralsar-Peshka made, he didn't say how you're supposed to implement that. It's more of a qualitative argument. Maybe something like this is what could be happening. Mm-hmm. So, but you're totally right. There's no clock here, and I'm not going to invent one either, actually. So that's an open problem.
Mm-hmm. Thanks.
Okay, let's come back at 8:55.
All right.
Or that's 8:55. What local time here? So.
We'll see you in a bit.
We're back. Shall we restart?
Okay, let's restart. Are there any questions on this first part of the lecture at this point?
Yeah, you've been extremely clear. So,
all right, so then I'm going to continue with the second part. So what I want to do is sort of reinvestigate some of the questions that I just brought up. But what I want to do is, take a more population-centered view on those questions. And the motivation in some sense is what we were looking at on Tuesday's lecture, that if we look at neural populations, across different areas of the brain, there are like several things that we find. So one is, activity is usually low dimensional, meaning that if you perform linear projections of neural activities, they eliminate the kind of diversity and complexity that you find in single cell tuning in favor of a simpler population picture. Then we found demixability or orthogonality. The different task parameters are represented along orthogonal axes in state space. We found distributed codes, meaning that each of these projections is made up by combining many individual neurons. And these three principles, they may be best explained by saying that really they all follow from this idea that what matters are linear readouts, are linear projections of the neural activities. So what I already showed earlier, we have a bunch of spike trains, we filter them the way a synapse does, and then we have a linear mapping, and that linear mapping can be something like a projection, and then we get linear readout. And the low dimensionality here simply means that the number of neurons that enter here is much larger than the number of readouts that you have in a particular network or a particular population. That's why the activity would be low dimensional. And the demixability or orthogonality just means that different of these readouts carry different types of information. So maybe one readout would have sensory information, another readout would be having motor information, etc. And the distributed nature of the code simply means that this linear mapping D is pretty much random, okay? So all of this can essentially be explained by just assuming that the brain uses linear readouts. And again, we can think of the linear readout as a conversion of a binary signal, a digital signal, into an analog signal. And we can maybe argue that as a field, we really have understood how to do that. We have understood how to go from spike trains and extract analog signals. I think it's most beautifully exemplified in brain-machine interfaces, where people would maybe record from spike trains in the primary motor cortex. Then they do more or less this transformation. They get analog signals out. Those analog signals, they project into a screen. So maybe you take, your 100 neurons from M1, and you create a two-dimensional linear readout that you create as the experimenter. Okay, you create that linear readout, two-dimensional, and you make it a pixel on the screen. And then what the monkey can learn over time is to move that pixel with his thoughts. Okay, it's just thinking about moving the pixel, and it actually goes through the brain-machine interface. And the question I want to ask again, because I already asked it in the first half of the talk, is the thing we have not understood as a field is how to go from analog signals to digital signals. How do we go from linear readouts to spike trains? And that is at the core of the problem with the standard model, because in the standard model, we try to do this for a single neuron, and it doesn't work. Because for a single neuron, if you do this, you get a regular spike train, okay? And I'm going to argue that maybe the solution lies in not thinking so much about single neurons, but thinking about this as a problem from the population point of view, okay? And so then the question is, what does it mean to think about this from the population point of view? Well, the problem is we have analog signals here. We want to convert them into a whole bunch of spike trains such that we can read them out in the next stage, okay? That'll be sort of the cartoon scenario that I want to go through with you. And I'm going to argue that the reason this is an interesting scenario is because it's going to lead to many interesting explanations for what we find in action neural circuits. It won't be perfect, okay? There'll be lots of stuff missing, but there's some features that we're going to get just by looking at this simple problem. So I just argued that we're going to do this for a population, but for pedagogical reasons, we're still going to start with a single neuron, okay? And so the question we want to ask is the following. Imagine you are that neuron in that population. You are neuron number I. You receive two inputs, x1, x2. It's a cartoon scenario. Those inputs, they can change over time. So they're analog inputs. A bit like currents that you feed on your dendritic tree. And now what you have to decide is whether you should spike or not. Here's the readout. So whenever you spike, you're going to influence the readout here, x1 hat, x2 hat. Your task is to make sure that these two readouts are as close as possible to do these inputs. So imagine this is what has happened in the past. So the readouts have already changed. And now we are at this dotted line here at this time point. And now we have to ask the question, should we spike? Okay, that'll be our question, should we spike? So the first thing that we have to wonder is, what happens when we spike? So when we spike, we're going to send a spike down this axon here, and the spike is going to change readout x hat 1 and readout x hat 2. It's going to lead to an immediate change of those readouts. That's illustrated here. There'll be a jump of readout x hat 1. That jump is exemplified by some parameter, d1i. And then there's another jump in the second readout, which will be exemplified by another parameter, d2i. Those two parameters, d1i, d2i, they make up a vector, a two-dimensional vector, which I'm going to call di, which I'll call the decoding vector of that neuron, okay? Whenever that neuron fires, the readout is going to change in this direction. So I should first explain, this is the signal space. We have x1 and x2 on the x and the y-axis. The signal is up here. That is the value of the signal at this particular time point. This is the value of the readout at this particular time point. And when the green neuron fires a spike, the readout is going to change in this direction, okay, in the direction di. So now the question is, should we spike or not? So if x hat is down here, then I would say, yes, the neuron should spike because the readout is going to move closer to the signal x, okay? And our goal is to be as close as possible to the signal x. On the other hand, if the readout is up here, so the readout is up here, then now firing is not such a good idea, because if we fire now, we're going to move to this point, which is further away from the signal x, okay? So not such a good idea. And by these kind of geometric arguments, we can sort of see that, any point in this space up here, the neuron should not fire, but any point in this space down here, the neuron should fire, okay? And then we have this green line here, which in some sense is the threshold of the neuron, because it distinguishes the non-spiking from the spiking area. Now, when neuron 1 spikes, so when, for instance, the readout reaches the threshold, the neuron will spike, and then it will reach another line, which in some sense is we're going to call the reset line. And the words threshold and reset are supposed to evoke the idea of a voltage, because this is a signal space, so it's very abstract, so that we can maybe think of these lines that are parallel to the threshold and the reset lines as the voltages of the neuron. Meaning that as we move in the signal space in this direction, the voltage of the neuron actually increases, goes through the reset, gets bigger and bigger, reaches the threshold, and now it's above threshold, okay? Mathematically, the voltage is simply the projection of the error between the signal x and the readout x hat, so the difference between x and x hat, on the decoding vector of the neuron di, so this vector. So an inner product between di and x minus x hat. Biophysically, we can think about it the following way. So we had our neuron that received the inputs x1 and x2. And what we now saw is that what we need to do to be able to answer that question whether we should spike or not is we need to take the readouts x1 hat, x2 hat, and just subtract them from the input. Okay, so I'm subtracting x1 hat here, I'm subtracting x2 hat here on those 2 bits of the analytic tree. And now what is being fed into the neuron is no longer the input x1 or x2, it's now the error, the difference between x1 and x1 hat, and the difference between x2 and x2 hat. Okay. So this way now the neuron will just see what's the size of the error. And if the error is big, the neuron will fire. And I can sort of show that in a little simulation. But conclusion number 1 from the assumptions we made is that if we want to build a system like this, that does kind of like an analog, digital analog conversion, then the voltage is going to measure something like a coding error. Okay?
Can I ask a question here?
Yes.
So do we assume neurons have memory of X1, X2, so they can compare?
They don't necessarily need memory. So what we're going to assume is that this is very fast. Okay, the spike goes through here, and x1 hat is basically immediately subtracted here. So then, at a time point t, you see this input x1, and you also see, x1 hat has a little bit of a delay, but the delay is going to be very short. Okay, very short, less than a millisecond. Let's say 0.1 milliseconds for mathematical convenience, and we can come back to that question later. So imagine the delay is very, very short. Then you immediately only receive the difference between x1 and x1 hat. Yeah.
Okay. And I have a second question here. So why do we fix the direction of the DI? Like it can only point to the reset, but cannot directly point to the X.
So the key is that the direction of DI is always the same because the readout, the contribution to the readout is fixed. Okay. That follows from our definition of the readout. So these parameters, D1i, D2i, they're given beforehand. The neuron's contribution to the readout is always the same. That comes from the definition of the linear readout as a projection. And since it's always the same, the vector points always in the same direction. No matter where you hit the threshold, the vector always points in the same direction.
Yeah, thank you. Yeah, that's clear. Thanks.
So there was one more
Claudius?
question that got solved.
I think Claudius has a question.
Yeah, I had the same question. If the readout is fixed in your model, but you just answered it. So thanks.
Exactly. The readout is fixed, basically.
And you will not learn it or update it. It's a fixed by before you start this.
Exactly. It's fixed before we even start it. And the reason it's fixed is really this, okay? That we say, when we do this with the data, it's fixed. If you do it for a brain-machine interface, it's just a fixed mapping D, actually. And then truth is, in the data, if you have your brain-machine interface, for instance, you run it over multiple days, then you actually do have to update D, okay? So it's not that fixed. But on the time scale, of hours or minutes, in the data, at least, in real neurophysiological experiments, the readout will not change. So that'll be our assumption. And you guys are asking a very good question. Where do you get this D from? Well, D for now is an assumption that we put into the model, okay? So assume that for some magical reason, I already know there's this readout D, and I know the direction of it. So now I can simulate this system. So here I have my inputs. The inputs now change over time. There are 2 inputs. Then the neuron, generates spikes. Those spikes change the readout. So you see every time, the neuron spikes, the readout sort of bumps up, both the readout x1 hat, but also the readout x2 hat. And then this readout is being subtracted from the input. So what the neuron actually receives is the difference between the input and the readout that's being summed on the 2 branches of the analytic tree, and that gives rise to the voltage. Okay, so voltage is the projected error now. And that you see, and then if the voltage goes up, that's because the readouts here are dropping. The voltage depolarizes, reaches a threshold, the neuron fires, the readout jumps up, and as the readout jumps up, the voltage jumps down, okay? Now there are 2 more observations about this system I want to get through. I'm going to show some movies, which I hope clarify or give some intuition on how this works.
Christian,
So,
can I ask you a quick thing? So here I understand it's a simple example that is illustrative, but in general, what I would like to generate is not exactly the input to the neuron, but some computation that is relevant for behavior or downstream processing. And so I would have to back up that quantity that I want. I would have to transmit this error all through the network.
No, I think, so there are two ways of thinking about this. So one is the simple way, and I'm going to try to argue this. So the simple way is that I just told you X1 and X2 are kind of input currents, okay? And all we're trying to do is we're trying to convert those input currents into spike trains so that a downstream readout can, read them out again in some sense. And we're going to the population level. If you wanted to put computations into the system, you kind of could try to put them before that. Or biophysically speaking, you could just say, well, I'm going to build an ADA converter that basically starts with maybe the input currents into the soma, generates spikes, then you have readout, and then you have a dendritic tree, for your freedom to implement whatever computation you want. Okay? So all the computation is now done in the dendritic tree. And the spikes are purely there to transmit the result of those computations to the next layer.
Okay, so then you assume that all the relevant computation is done in an analog way in the dendritic tree, and the spikes are just there so that the downstream neuron can kind of recover what was in the dendrite.
Okay, that's version 1 of the story. For version 2, you can ask me at the end of the talk again.
Okay, thanks.
But that's the version I'm going to try to explain here. Mm-hmm. So there's one more thing I want to point out, and that is, I've shown you this kind of architecture, but there's really only one neuron. And you also see that, when the input changes, actually the readout doesn't change at all. And that is because we're trying to reproduce a 2-dimensional input with one neuron. So that can't work. So we will need more than one neuron. So I'm going to introduce a second neuron. And I'm going to assume that the readout weights of this neuron are different. So it's not an identical reproduction of the first neuron, because its contribution to the readout X1 hat and X2 hat is going to be different. But when its contribution is different, it'll have a different threshold in the signal space. Okay, the threshold will be like this. And then I can introduce 3 more neurons, shown here, and they all have different contributions to the readout. And I set them up, and this is all just done by design, in such a way that they now bound the input signal such that the readout cannot leave this white space here in the middle, this pentagon, okay? The readout will no longer be able to leave this pentagon, and thereby I've now produced a system that basically is able to convert that signal into a bunch of spike trains so that I can read it out with a defined precision, which is given by this pentagon. And I'll show, if this is a bit too much, I'll show a movie that hopefully clarifies that. What I want to point out is that we now have 5 neurons. They all do exactly the same thing, except that they have different contributions. So they have different decoding vectors. That's why they get different voltages here. And you see that now, some of these other neurons are firing, and thereby the readout is now tracking the inputs. Before showing you some simulations, some movies for this network, I want to point out 2 more reasons why this is an interesting way of thinking about it. So one thing that you may wonder is, what on earth is this thing that this guy calls a voltage? Like, it's a projection of the error. That's not a voltage. Where's this all supposed to come from? But it turns out that this is not so far away from something that may be more familiar to all of you. Because if I take the readout X hat and I plug in its definition, okay? So I'm not going to go through the math. I'm just going to show you the results. If I plug in the definition, I can expand it in the following way, where I have a sum over all the other neurons in the network, all the neurons J, and I get these terms, which are inner product of the decoding weights of two neurons, okay, the decoding vectors of two neurons. So DI inner product with DJ. RJ is the filtered spike train of one of the other neurons. And these inner products have the form of a synaptic weight. So I can reinterpret my network without changing anything. It's just a reinterpretation of the math as saying that I have lateral connectivity here. So I don't actually need the readout and feed it back. I can do everything through lateral connections in some sense. And then if I now take the temporal derivative of the voltage, again, I'm not showing this, I'm just giving you the result. What I actually end up with is a network of leaky integrated fire neurons. So this is the temporal derivative of the voltage. Then there's a leak. There's a feed-forward input, which has the signal X. And there is this term here, which are recurrent inputs from all the other neurons, okay? So it's not that the voltage was like a weird thing to start out with. It's actually just an integrated fire network. The only thing is that it has a very special connectivity. The connectivity is set up in such a way that we obtain this bounding box, okay, that bounds the error. The third point I want to point out, the third point I want to make is to just look at this yellow neuron here. So this neuron. So this yellow neuron is actually not firing any spikes for this input, okay? But you still see that the voltage of the yellow neuron changes. In fact, you see that it receives a sort of excitatory input because its input increases. And then there's a bit of a hyperpolarizing decrease, okay, an inhibitory input. And then there's a depolarizing excitatory input and the hyperpolarizing inhibitory input. And what you can see is that each bit of excitation is exactly matched by an equal bit of inhibition. What does the excitation tell you? The excitation tells the neuron that the error is increasing, and then the inhibition comes actually from the green neuron spiking, and it tells this neuron, well, the green neuron just took care of the readout error. So that's why you need to reset. Then the yellow neuron has reset. It sees that the error is increasing again, and then bang, it gets inhibited by the green neuron again, because the error was just eliminated by the green neuron. And so this neuron is actually balanced. It's in a state of excitatory inhibitory balance, because here, each bit of excitation is exactly eliminated by an equal bit of inhibition. So it's a kind of tight balance, basically. And now, balance is no longer that thing that we need to generate random spike trains. In this network, there is something else. It just illustrates that excitation creates errors, inhibition eliminates errors. For all errors to be eliminated properly, excitation and inhibition have to be balanced. That's what balance means within this type of framework. The proper elimination of errors. So at this point, I usually just like to show some movies, because I imagine that, this may all be a bit confusing. So the movie I want to show is how the network tracks a sine wave and a cosine wave, okay? So if you have a sine, and so here's the signal space. This is signal x1, this is signal x2. And what I'm going to do is I'm going to have a sine and a cosine. So they go like this, okay? The sine and the cosine in signal space goes like this. Here, I'm going to show you the signal and the estimate. So the signal is going to be a cosine and a sine, okay, over time. So it's the same thing, it's the signal over time, okay? All right. And so I'm now going to start the movie, and then I'm going to show you how that thing is being tracked. Okay, so we start with the sine and the cosine. So this is the signal, it'll go around in a circle. Here's the sine wave, this is the cosine wave. And now I'm going to turn on 12 neurons that are supposed to bound this signal. These are the 12 neurons. The black dot is now the readout. That's the readout. The readout always wants to leak towards zero. Okay, it's trying to reach zero. In the absence of spiking, it'll reach zero. But it can't reach zero because as it tries to reach zero, one of the neurons' threshold is going to be hit, and that neuron is going to fire a spike, okay? So I just zoomed in so that we can see what's going on inside this bounding box. This arrow shows now towards zero. And you see every time one of the neurons' threshold is reached, the respective neuron is going to fire, and it's going to, put a spike, and that spike is going to change the readout. And as you can see in the simulation, the black line is now tracking the gray line. So we've built this digital analog converter, okay? We've built a system that takes an analog input, transforms it into spike train, such that you can read it out afterwards. The readout is showing the black line here, okay? And the precision of this representation is simply given by the size of this box. So if I make the size of this box smaller, I can get arbitrary precision. By the way, did I freeze? Because I think my computer has problems with running movies and doing Zoom. I did freeze.
I think you're sort of okay, but it's rather slow.
Okay.
It's a little choppy.
It was a bit choppy.
Yeah.
I apologize. I think it's not my internet, it's my computer, because I noticed that the movie was kind of choppy. So let me show the movie again. I was trying to explain things as it went along, but maybe I just briefly explain it. So I'm going to show the movie again. So the gray line here is the sine and the cosine. Those are the two signals. The gray dot is the signal. The black dot is the readout. The readout is always going to try to reach 0. Okay, it's always trying to reach the origin of the coordinate system. This arrow here shows where the origin is in this case. The readout cannot reach 0 because it's going to hit the boundary of one of the neurons. Okay, that neuron is going to spike, and that spike is going to change the readout, and then the readout jumps back into the bounding box. Okay, that's the essence of it. So let me show this to you again. So this is the signal. Okay, is the movie at least coming through?
Yes.
Yeah, not being choppy. I just shut up so that at least the movie comes through without being too choppy. Okay, so that's the essence of the key ideas. And I don't show you the voltages of the neurons, but you have to believe me, this is just an integrated fire network. It's just a particular way of illustrating what's happening. So why is this interesting? So there are two more reasons why I think it's interesting. So one of the things that I started out with saying is, we don't understand where the variability comes from in the nervous system, all these noisy spike trains. So the next thing I want to illustrate is, one way of thinking where all that variability comes from. And I'm going to show this with another movie, and I hope it's not going to be too choppy. But if it's choppy, you guys should stop me. So what I'm going to do is I'm going to show you the same thing again, but here is signal X1 and X2, except I have more neurons now. Okay, this is like 25 neurons. So they form this type of bounding box. And here I'm going to show you a signal that is a bit different. So the signal here will just be a constant signal. Okay, it's just going to be a signal like this. It'll just be a constant signal. And then here, I'm not going to show you what the whole population does. Rather, what I'm going to show you is what the red neuron does. Okay. So here is a red neuron that is a bit highlighted. It's supposed to be a bit thicker. I don't know whether you see that. So I'm going to show you on each trial the spikes that the red neuron fires. Now, in a completely deterministic system, it'll of course fire spikes exactly always at the same point in time. What I'm going to do though is I'm going to introduce just a little bit of noise in each neuron, okay? Not a huge amount of noise, just a little bit of noise. And I really just need a tiny bit of noise. And I'm going to visualize the noise for you. So what the noise is going to do is going to move around those thresholds. Because what happens is if there's a bit of depolarizing noise, the neuron comes closer to threshold. And I'm just going to show that by moving the threshold closer to the readout, okay? It's just a visualization of the mathematics. And so let's look at that. So you see the thresholds are all moving a little bit. And this is the readout as it moves around. And I'm only showing you the spikes of the red neuron. And so what I hope got visually clear here, yeah, I know this is a bit jaggy, is that you see that, you know, there are very complex trajectories of the readout inside this bounding box, okay? Nonetheless, the readout does not leave the bounding box. So the signal here is always being tracked with basically equal precision, okay? The precision with which we track the signal doesn't change. However, because there are many possible ways in which the readout can hit these boundaries here, because of the little bit of noise we added, the red neuron now, you know, changed on each trial how it contributed to the readout. And that's illustrated by just counting how many spikes that neuron fired in each trial. So in the first trial, it fired 5 spikes, then 7 spikes, then 2 spikes, then 6 spikes, etc. So you see there's a lot of variability in the firing pattern of this red neuron. At the same time, you know, because of the geometric construction here, that the population readout wasn't noisy at all, okay? Well, strictly speaking, it has a little bit of noise, but it's very small noise compared to the variability that you see in the single neuron. So why is this happening? So the key message here is that basically there are many possible spike patterns that this population of neurons can generate to represent that input signal. There's not just only one spike pattern, there are many spike patterns. There are many spike patterns because there's redundancy in the code or degeneracy in the code, because there are a lot more neurons than there are linear readouts. And because of this degeneracy of the code, what the system is going to do is in each trial, it finds one of those population spike patterns. And in the next trial, it finds another population spike pattern. And in the next trial, another population spike pattern. But if you only observe the red neuron, you know, it will start to look very, very noisy because you're not seeing the full picture. Even if you saw the full picture, it will maybe be hard for you to actually see what's going on because you need to know what the readout is, the proper readout, to actually be able to, you know, get rid of all of the noise. So that's one observation. So even if there are no small differences in the noise in the individual neurons, so even if there's very small noise in the individual neurons, there can now be very large differences in the spike trains of individual neurons, okay? But those are just a reflection of the fact that what we have to do is we have to look at the population, not at individual neurons. Only the population actually tells us what's really going on. The last point I want to make about this framework is one about—
question before you move on.
Yes, please do.
I probably need to ask this a way ago. Why do we have the minus to the recurrent network in your equation?
So why is it minus here?
Yeah.
So the minus simply comes from the fact that we're trying to compute an error. So here we have an input x, and we're trying to subtract we're subtracting the readout x hat. And so that gives us the minus, okay? And then when you go to the math and do a network of leaky integrated and fire neurons, you receive a minus here. And maybe the easiest way to think about the minus for now is to just think that these connections are in effect inhibitory. It's not strictly speaking true. I can give as a homework why that is not true. But I think it's the easiest way of thinking about it, that basically the neurons are inhibiting each other. So neurons that have similar tuning, similar inputs, they will inhibit each other. They will try to, you know, avoid firing for the same input. So if one of them fires, it'll inhibit the other one to say, don't fire, I already took care of the input. Yeah.
But we know among excitatory neurons, they do have recurrent network that they kind of enhance the signal.
Yes, that's right.
Just because, so we don't discuss that kind of situation here. Right?
Yes, and I'm not discussing that kind of situation. Excellent, excellent point. But I will come back to it later, okay? You're totally right.
Thank you. Yeah.
This is still just a cartoon network. So there's one more thing I want to show about this cartoon network that I think
I
is.
got a question about
Yeah,
that page. What about the number of the neurons increase, the noise differences? Will it be decreased as the population goes up?
Can you repeat the question?
My question is, as the population of the neurons you put into this network, will the noise have less impact on the result?
So that's a good question. So that's a complicated question. So it turns out that if you increase the number of neurons here, okay, then the noise will actually have a larger impact on the readout. And that is because the neurons that affect the readout are always the ones that are driven towards the inside of this bounding box, okay? So the larger the system becomes, the more sensitive it becomes to noise, which is a bit counterintuitive. It's one of the problems of this system.
A similar question that I may have is, what is the noise correlation in your network?
The noise correlation is if you scale it up, okay? So basically the noise correlation should be that neurons that have similar tuning have negative noise correlations, okay? The problem is you can only see that in cartoon networks. Once you have very large networks, so networks that are large, not just in the number of neurons, but also in the number of signals they represent, you won't be able to see that anymore by looking at pairwise neurons. And then the noise correlation is effectively zero. Yeah.
Yeah, I kind of think this kind of a problem, because in my assumption is that a brain has so many neurons, they can kind of narrow down the noises impact. Is that true, or is this on the contrary?
So I still have problems understanding you acoustically. So I understood that you said the brain has so many neurons that something with a noise, but I didn't understand it. Can you repeat?
Yeah, I mean is that the brain has many neurons, so each neuron will have a big impact on the different on the noise. So we have so many neurons, we can narrow down the noises, the to impact output and the readout.
Right. So you think that we have a lot of neurons to eliminate the noise, basically. That is an assumption, I would say. It's possible, but it's also possible that it's not true. Okay. It's possible that the architecture works in a way in which noise is eliminated in a different way, not just by averaging over neurons, for instance. So I'd say it's an open question. I would also want to say something else about noise, just while we're at it. So noise, of course, is prevalent in all of our experiments, right? We basically measure trial-to-trial variability. We observe, shared noise covariations and activity and all that kind of stuff. It's really important to keep in mind that first and foremost, the noise that we measure is really experimental noise. Okay, so quite often we assume that the noise that we measure is also the noise that the organism has to deal with. But that, of course, is not true. The point of view of the organism is a different one from the point of view of the experimenter. So just because you measure some noise doesn't mean that a downstream area is measuring the same amount of noise. It could be less, it could also be more, right? So it could be that from the point of view of the brain, there's more noise than in your experiment, because you assume things that the brain doesn't know about. For instance, you assume that all of these stimuli are the same, but maybe from the brain's perspective, they're not the same. Okay. So it can be either way. And often we have a tendency to just think, our measurements are correct. And the noise that we see is also the noise that the brain sees. But I think that's generally misguided. So we have to be very careful to make that equivalence by saying, if I see variability in my recordings, the monkey must see the same amount of variability. Okay, we don't really know that. Yeah. So let me continue maybe, and then we can come back to some of these questions later. So there's one more point I wanted to make, because I promised you something about robustness. And that is the movie I wanted to show exactly. So one thing I started this talk with is, how do you build a system that is robust to eliminating neurons? And so I want to show that this system is actually robust to eliminating neurons. And you can see that in this simulation. So I'm showing the same simulation as before. We just look at the red neuron and its spike trains. And what I'm doing is I'm killing 50% of the neurons. And then I'm just speeding up the simulation. So after I killed 50% of the neurons, you see there are a lot less neurons now. But you can also see that, the readout still is contained inside this bounding box. Because the key insight here is that if you eliminate a couple of neurons, it doesn't really matter because the other neurons can still take care of the error. Okay? So you have less neurons here, but there's still an intact bounding box. Okay? The bounding box is still intact. You can kill even more neurons, which I'm going to do in my next simulation. So I'm killing 20, I kill more neurons, another 50%. And you see now that even with these 7 neurons that are left, or 8 neurons actually, 8 neurons that are left, I'm still able to completely represent the input signals. So I don't really need all those 25 neurons, okay? And that in some sense is the essence of why the network is robust. Because there's a way in which the information is represented that is does not rely on single neurons. It's a population code in many respects. And the only thing that you need for the system to work is that the error is bounded in all directions. Okay? As long as that is true, you should be able to represent the signal.
Sorry, Professor.
Yeah.
I noticed it seems that less neuron, the spike time interval seems to be smaller, like the frequency of the spike.
Right.
So
Yes. So that's an excellent observation. So what we see is that now that we have a lot less neurons, each of these, the red neuron fires a lot more spikes than initially. So now fires 18, 19 spikes. The other thing, so it fires a lot more spikes. The other thing we observe is that the variability has disappeared, okay? There's a lot less variability now. You have like 18 spikes or 17 spikes, 19 spikes, but the initial variability is sort of gone. And the reason is that by eliminating so many neurons, we've eliminated the degeneracy of the code, okay? There are now a lot fewer spike patterns that can actually represent the signal. And therefore, because there are fewer spike patterns, there's less overall variability in each individual neuron. And you should be able to see that on the level of an individual neuron. So that is something that we're predicting, and we're actually now currently testing that with a colleague of mine, Alfonso Renard, who I managed to convince to test this particular prediction. But of course,
I have a, sorry,
yeah.
Could I ask a question again? I wonder what's the criteria for killing neurons? Is there any, like, for example, if you just kill like one side, eliminate them completely, and there's no bounding box anymore, right?
If you can kill them all, then there's no bounding box. That's right. But
no, I mean, if we, yeah.
You may ask the same question that this guy is asking, which is Ramon y Cajal, or just, someone that comes more from the neuroscience side, and it's like, okay, neat theory, but which neuron shall I actually knock out? And the problem is, we have this little cartoon model, but if you now go into a real biological circuit, such as cortex, and then you say, well, we have pyramidal cells, we have all these different types of interneurons, basket cells and chandelier cells, et cetera, Martinotti cells. Which of those neurons am I supposed to kill? Okay. And so the short answer is that there's a big mismatch between this cartoon theory and the biological reality. So what are you supposed to do now as a theorist? Okay. So you can say, well, I have a theory that has several intriguing possibilities. It has several intriguing explanations for, variability, for balance, okay, for robustness. So those are things that are all sort of attractive because they explain certain features of a real system, but there are also many features that we're not explaining at all. And a big problem to start out with, now before we go to all of these different neurons, is that our neurons have excitatory inhibitory connections, but they do not have excitatory inhibitory neurons, meaning that each neuron can send an excitatory, but also an inhibitory connection to downstream partners, which is obviously not the way it works in a real system. Now, in all fairness, if you look at our functional networks, like deep networks, they have the same problem, okay? They would also mix these things and not care about it. So as a field, I think there's a deeper underlying problem, and that is we actually do not know why Dale's law exists, okay? Why neurons separate out into excitatory and inhibitory neurons. So it's an open problem currently. Other people are also interested in this. And so there are indeed several steps that are missing from this, ADA converter to a heuristic network. And so that is, ongoing work in the lab. The way I think about it is that a framework like this is not really just a theory. It's more like a path that you go down, okay? It's a path that may lead to something interesting or maybe not, right? It's a path that has already led to some predictions. We're trying to test those predictions because, of course, one nightmare for every theorist is, you work on some theory for, say, 20 years, and then afterwards some experimentalist comes along and says, well, here's the experiment that definitively disproves your theory. And then you can say, well, I just worked 20 years on the wrong thing. So we're also trying to sort of see, do the predictions that we think are essential to our theory already hold. But there's more work to be done. So, this is something that we have done now, and there have been work from other people in the field also. There's been some nice paper by Sengic Pilavan, for instance, where the question is, how do you match this onto excitatory and inhibitory neurons? So I'm going to skip this here. That is not super important. There's one more thing I want to show. There are two more things I want to show. So one is just a demonstration, even though my movies are not working very well. So if you have more than two signals, what happens? Imagine you have three signals that you want to represent, okay? So if you have three signals that you want to represent, then your signal space is three-dimensional. And then each neuron still is a bound in that signal space. But a bound in a three-dimensional space is a plane. So each neuron's threshold becomes a plane. And if you put all of these planes together, you get this weirdly shaped object, which is still a bounding box. And now the readout moves around inside this three-dimensional bounding box, okay? And now maybe this will be too jagged. My computer cannot do this. And you see that we're basically tracking a three-dimensional signal, okay? So that is what happens in 3 dimensions. And then you can imagine you can scale it up. But of course, if you scale it up, you cannot visualize it anymore, okay? And the last thing I want to illustrate is maybe this one here, because it's a beautiful experiment. About the question of instantaneous recovery and robustness in the brain. So this is a nice experiment done in David Dupre's lab in 2016 in Nature Neuroscience. And they did the following thing. They had a mouse run around in an arena like this. Now, if an animal like a mouse or a rat runs around an arena like this, we know very well that we can find place cells in the hippocampus. And so here they show 3 place cells. So this is the circular arena, and you find a cell that is firing whenever the animal is in the left bottom part of that circular arena. And here you have a cell that's firing whenever the animal is in the left part of the arena. Here's a cell that's firing whenever the animal is in the right part of the arena, okay? Now, by some kind of crazy molecular techniques that people use these days, they were able to tag those cells that were active whenever the animal is in this arena, okay? So they tagged these place cells. They tagged them, and by tagging them, they expressed an opsin that allowed them to shut down the cells with light, okay? And so what they could do is they could just shine with a laser some light into the hippocampus and turn off these place cells. So imagine one of these cells is firing, then the laser comes on, light comes on, and the cell basically turns off. And this is what happens when the cell is turned off. Well, the light is turned on and the cell is shut down. And you see the place field has disappeared, obviously because you're silencing the cell. And then the second cell, same thing. You know, it had a place field in its normal condition when you shine the light and you inhibit the cell, because that's what the light does, it starts inhibiting the cell, then you know the place cell is gone. And here too, the place cell is gone. And now the interesting observation they made is that there were previously silent cells. Okay, so these did not get tagged. They were not very active when the animal was in the arena in the normal condition. But as soon as you turn on the light, where as soon as basically, you know, within 1 second, if the animal is in the right spot, those cells became place cells. So there's something like an instantaneous recovery of the place cell code. So even if you take all the cells that were active in the hippocampus in this arena and you shut them down, there's almost immediately another place cell code, which I find is quite an amazing experiment, that the place cell code just comes back, okay? And so that is something you can also understand with the idea of this bounding box, because if you imagine, you know, that you have cells that are all active, these are the cells that are active. And then let's say there are some cells that are a bit more hidden, okay? Maybe more in the corners, so they'll be less active because, you know, they don't get hit that often. They don't have so much exposure. Then if you now eliminate all the black cells, okay, then what's going to happen is that the orange, oh, sorry, I'm screwing this up. This would work better on the whiteboard, I'm sure. That now the orange cells will become active when you eliminated the black cells, okay? So that would be our explanation for this type of representation. Okay.
But I think this also depends on the behavior that you see, right? So basically whether the optogenetics will change the behavior of the animal, because there are recent papers by Michael Hauser's group, right? They can do calcium imaging and optogenetics at the same time. And they show that if you do, the mouse are doing this kind of experiment and you find these cells, then you activate them or inhibit them, it could impair the behaviors, if I remember correctly, right? But in your scenario, it's like redundant code.
It's a redundant code. So what you're asking is, did the behavior change?
Right, right.
And so the claim of these authors is not really actually.
Yeah,
so
your prediction is that, yeah, your prediction would be it's not going to change, right?
Right, that's right. That's true actually, yes. Our prediction basically is that if you randomly knock out some neurons, and in this case, you know, you are not knocking out all of the neurons, you're only knocking out a certain percentage, that the behavior will not change. That's right. That is the prediction. So all of this is sort of, you know, I think intriguing evidence that, you know, we may be capturing something of what the brain does. But obviously, there are also many things we're not capturing. And I think that also came up yesterday in the question of, you know, theory. All theories are wrong in different ways, okay? And so there are many sort of competing theories that capture some of the other aspects much better of what the brain does. And so I guess the task for all of us will be to sort of try to expand the realm of what one theory can explain. And maybe sometimes they get merged or something new comes out of it. And then maybe eventually we'll have something that, you know, approximates what real circuits do. So with that, I want to end actually. So my assumption, the key assumption that I made in this network we just built is really that, you know, signals can be read out linearly from spike trains. We just assume that these decoders are random, basically. And spikes are only fired when that's absolutely necessary to keep a readout in check. And well, there's redundancy in the code. So those are the key assumptions that we made, okay? And then the key results were the membrane voltage represents a projection of a coding error. Neurons are coupled leaky spiking units. Whether neurons exhibit EI balance or not is a question of dimensionality or redundancy that I didn't really explain, so it doesn't matter. But then I had some predictions, so that randomly killing or silencing neurons in the network will increase firing rate, increase regularity of firing, and decrease trial-to-trial variability. And the last thing I also skipped over. So thanks. You know, ask me any more questions if you have more questions.
So, I mean, I have a question about maybe some of the students can go first.
I do have a general question here. I'm trying, all through the lecture, I'm thinking like, why do we killing neurons? And you mentioned about how the like dementia or this disease we can apply on. Can I also understanding in a way say we're trying to find this fundamental network or function of the neural network? Like what's the most necessary network?
Yeah, I mean, there are different ways of thinking about this. So I didn't really show you how to do any computation, right? I only show you how to go from analog to digital to analog. So first about what you said, we should be very careful because there's no evidence at this point that networks are actually robust in the way that we propose to the knockout of neurons. So this is some evidence that networks are robust, but that doesn't mean this robustness comes from the type of mechanism that we show. Okay, there could be other ways in which you can think about this. You know, maybe these place cells are just being tagged because no one else is. They just are being regenerated by plasticity, say. Okay, so there's always the possibility that there are other mechanisms that make the network robust that are just plasticity. And so I don't say anything about this. So the idea that it's not plasticity, it's the code, you know, that has not really been proven experimentally. So just a hypothesis. And the way I would like to think about the theory is something that I said yesterday. I think what's important in a theory is, you know, what's the amount of assumptions that you make, and what is the amount of, and what is the complexity and the amount of stuff you get out. So we obviously are lacking a lot of things in this theory. On the upside, we did not make a lot of assumptions, okay? And so if you think that something's wrong here, you'll have to basically think, oh, which of these assumptions is wrong, right? Because everything just follows from these assumptions. So obviously we didn't end up where we wanted to end up. So something has to change here, okay? I can give you my hint on what I think is wrong. So I don't think that this is wrong. I think signals can be read out linearly from the spike trains because it works so well in experiments. I also think that, you know, the spikes are only fired when necessary. Seems to make a lot of sense. I think they're redundant. So I'm obviously not giving you the full assumption. There's also an assumption we put in about the decoder. Okay, we said the decoders are random. Okay, and I think that's probably not quite correct. So there are probably some constraints. For instance, one constraint that we have investigated in the past is the constraint that all the elements of the decoder are positive. If you make that constraint, then you actually end up with a network that is all inhibitory, okay, and has a bit of a different architecture. So I think this is the thing where the network that I showed today kind of failed, because we just assume that, that these can be positive, can be negative, they're kind of random. And I think that's something that is not quite correct. That also comes from the experimental observations, though, because when we read out linearly, we find that readouts are positive and negative. But there's something that I think doesn't quite map 100%. So this is where we're currently working on. We're sort of wondering, what is actually, that's a large set of parameters. So we start with random because that seems like the easiest thing to do, but maybe that's too simplistic, okay? Alternatively, if you say, I want my decoders random, you have to cut something else. So you can say, I don't believe in linear readouts. I think that's false. You can say, I don't think that spikes are fired so efficiently. You can say, there's more noise in the system. This is all just a lot less efficient than you think. So many people will say that. Many people will say, oh, this is not as efficient as you think. That's a possibility, okay? You can say neurons are not redundant. That's a possibility. Then you would predict whenever you knock out neurons that there will be deficits. So the strength of a theory here is not so much that you do something and then you already have everything. The strength of a theory is more that it allows you to think in certain directions, and it'll also very strongly highlight what you don't understand. So for me, for instance, just to make this also a bit personal, before I started working on this theory, I never thought about Dale's law. For me, it was like, well, the excitatory neurons, the inhibitory neurons. So you start with that as an assumption, okay? So from a sort of bottom-up perspective, you would just say, well, let's just say there are excitatory neurons and inhibitory neurons. So then this is you say, I already know this, so obviously I start with that. But if you start with something, you're not explaining it. Okay, starting with something is not explaining it. So coming from this totally different perspective, I have a very different problem, because now the problem I have is I have to explain it. I didn't start with this. I started with the linear readout. Okay, I'm not explaining the linear readout because that's what I'm starting with. But now I have to explain Dale's law. Okay, so this is really what's crucial about theory. You start with something, you're trying to explain something else. We didn't start with excited inhibitory neurons. Now this becomes something that we have to explain. So we can say, well, maybe that's too difficult to explain. Maybe we should start with that, right? But then you have to explain the linear readout, right? Where does this come from? So I guess there are different ways of cutting through the field. And if there's anything that I think we as a field do wrong, is that we're not heterogeneous enough, okay? We don't understand the brain sufficiently well that we should all follow just one approach. I think there's a huge amount of value in following many different ways of cutting through the system. And that is also something, by the way, that became clear to me by doing this network, because one of the ways this network is actually robust is through heterogeneity. Different neurons do different things, okay? And this is also how biological systems are often robust, because different bits of the system do different things. If you have something like the coronavirus, there will be humans that are immune to it. They won't get it. And it's because everyone's immune system is a bit different. So biology always has this heterogeneity built in. And as scientists, we should also try to build that heterogeneity into the field, right? As long as we don't have a theory where we can say, we all know this is correct and we can follow this. Like physicists they can say quantum mechanics is correct, so let's follow this through. As long as we don't understand the system, there's a lot of value in having a lot of heterogeneous approaches, like different approaches, to try to understand it from different perspectives.
So let me ask my question now. I was looking at the equations, and I was thinking whether or not you can put ion channels into your neurons at this point. Was it that will help? Yeah.
Yes. Excellent question. So basically what you're pointing out if I come back to my equations here, what you were pointing out is that if I look at this model here, this is a network of leaky integrated fire neurons, but it's a current-based model. So the spikes here arrive as delta functions, and they are currents. Okay, the right-hand side are currents here. And of course, that's not how a real neuron works. The input to a real neuron is not a current, the input to a real neuron is a conductance, because the transmitters, you know, from the synapse come and they open conductances. And so you have a conductance change, and then that conductance change leads to a current change. And so you can ask, you know, what if we make this into a conductance-based model? And in conductances, then you have ion channels, etc. And I think the short answer is, well, you know, we've tried it. It doesn't improve anything. It only makes things a bit worse. And then the truth is, you know, from this perspective, from which we're coming, I have to say, we don't understand why they're conductance-based neurons, okay? I have to say neurons should be like this. So as long as I cannot put conductance-based neurons in the framework, I don't understand why we need them, okay? So it's again, something where I say, well, maybe there's something we do not understand, okay? It's again, something that now I have to explain. I have to explain you know, why are there conductance-based neurons? And we've thought about this at some point, you know, but we have never come up with a very good answer. I have to admit, okay? And in conductance-based neurons, there's also a big difference between excitatory and inhibitory inputs. So excitatory inputs, because they're often way above the threshold, so this has to do with reversal potential. So maybe I'm digressing now, but just to finish my thought, if this is the threshold of the neuron, the voltage threshold, and that is the reset, and the way we think about conductance-based input is through reversal potentials. And so an excitatory reversal potential will be very far up here. This would be the excitatory reversal potential. Whereas the inhibitory reversal potential is basically down here where the reset is. So again, there's a big symmetry breaking. Sorry, now Zoom is playing tricks on me. Okay, Zoom does not like what I'm doing. So let me see. Okay. Yeah, this doesn't look very good. Sorry about that. I started drawing and then the slides disappeared. So here you are. So the blue line is the inhibitory reversal potential. The red line is the excitatory reversal potential. This is threshold and reset. And what it means is, from the point of view of an excitatory input, since the reversal potential is so far above the threshold, excitatory inputs are actually very much like current inputs, okay? They do activate like current inputs. It's a very small difference between the conductance and the current input. But that's not true for inhibitory inputs, because the inhibitory reversal potential is here at the reset. So it makes a big difference whether you receive an inhibitory spike when your voltage is down here or where your voltage is down there. Okay, it's a big change. Like in a current-based model, you always jump down exactly the same amount. But in a conductance-based model, you won't. You only jump at maximum to the reversal potential. And so, yeah, we don't have a good explanation for that. One explanation could just be, you know, you're trying to save energy there. Why jump down much further than I need to, for instance? But, you know, this is just hand waving. So I should shut up about this.
So I think we're sort of out of time, but I think we should thank Christian for this excellent lecture and this, you know, also previous excellent lecture.
Okay, thank you very much. Thanks, everyone. You know, if you have questions that pop up to you next week, just write me an email. I'll try to answer.
Could you also send us your slides, please?
Oh, yeah, I haven't sent yet, actually. Yes, yeah. Of course, I'll send the slides. So the movies, I cannot send, but I'll send a link for the uploaded movies.
Yeah, I will download it from your Dropbox. Don't worry about that. Thank you.
All right, take care.
Thank you very much.
Thank you very much, Christian.
Thank you again. Thank you so much. It was great lectures.
Yeah.
Okay. And enjoy the rest of the school. Bye-bye.
Bye.
Bye.
Bye. So for some of us, this is the last session. So Nimi want to thank everyone for being with us this entire week.
Sorry to interrupt, but maybe we can stop the recording at this point.
Right.